Dr. Hoerndli, welcome to the podcast. Pleasure to have you on. I think to start, just tell me a little bit about yourself. Tell me about your research, your background, and really what the story that brought you to where you are right now.
Okay, yeah, thanks for having me on this podcast. I'm really excited to be here. So my background, let's see. Well, so I was born to Swiss parents, but in Singapore. So I grew up, I'd say half of my teenage years in Asia. And then that sort of formed my view of having lots of interest in lots of animals. I would say that one of the formative experience there is that we did a lot of snorkeling and scuba diving. And so I was, sort of involved in how nature kind of really played a big role in how I grew up. And so that biology got me fascinated at an early time point. And I moved back to Switzerland for college. And so I did all my undergraduate work in Lausanne, the city of the Olympics. So in Switzerland, in the French part. And there I basically, in Switzerland, you have a 4-year bachelor's system, but you actually have a master's. We call it a diploma, but it's basically a master's that's inbuilt into the system.
So after 4 years, you get both?
So you basically do a year of research, but it's part of that diploma. So you have 4 years of classes and then a year of master's.
Okay.
And I specialized in plant physiology, which I was not expecting at the beginning, but that's what I ended up doing. And after that, I decided that plant physiology was not what I was interested in. And I have got an anecdote with that that can come back later. But during that time, my grandfather developed Parkinson's disease on my mother's side. And one weekend I went to visit him. And I actually used this example in class that I went to see him and he had forgotten. He lived alone, had lost his wife 3 or 4 years before. And then he had forgotten to take his Parkinson meds, so dopamine. And, that when in the early years before I studied neuroscience, that was the first sort of wake-up call or realization that somebody he's a high school teacher in math. He was very smart. He could have been a professor, a university professor, but born just before the Second World War.
Yeah.
Was not an option. And so he basically showed me, when I saw him, he could not function. So I took care of him for 24 hours. And within 24 hours, with the dopamine medication I gave him, it was amazing, the transformation. You go from someone who's essentially almost in a vegetative state. So he couldn't talk. He was there, but only physically. And within 24 hours, he become a person. And that experience, that idea that our neurons and the chemicals that we can take can change our state, ourselves, that really triggered this deep need to kind of go into research.
That was like your moment of I need to understand this.
Yeah, I want to know, and I didn't know where. I didn't know if it was going to be like, psychiatry or medicine or where, but it was this, realization that there's this basic biology going on that if we don't understand, that can completely transform a person. And that really was the fascination.
Yeah, it's super, super interesting. I remember taking cellular neurobio, and it really blew my mind. I had no interest in neurology, neuroscience, any of that before I took your class.
Interesting.
And then after I took the class, I was like, this is the coolest stuff I think I've ever learned, because it's this black box, right? The brain. Everyone knows the brain, the nervous system. It's how you think, how you control your movements, and so on and so forth. But once you break it open, it's like, whoa, it's beautiful, really.
There's so many components. The brain itself is also sort of an allegory for things, that there's the diversity of the neurons that have to play together. And what's amazing, now being a parent, having 2 children, is also the realization of, especially when you teach a class like that, where you show examples of diseases or disorders or mental health or things like that, you wonder, how does it even get to the point where it actually works? And most of us work.
Yeah.
Right? Most of us function completely fine. And the answer is redundancies within redundancies within things. And, we think about the idea of like, you break apart something. And as kids, we all did this. I did this. I see my sons doing it. You break apart a toy and say, oh, yeah, how is it you put it back together? And of course you can't. But you kind of realize, whoa, this is more complicated than I thought, right? The parts fit together. They fit tightly together. And the same thing is true with the brain. there's so many different parts. And I think the one thing that really drew me forward after that was also the idea of research, right? So there's that experience was, okay, I'm interested in that, but why didn't I go into medicine?
Yeah.
Why did I decide for research? Well, one aspect of that is at least somewhat the acceptance that I couldn't really deal with the idea that if I did a mistake or had a problem with a patient, that person's life would be in my hands. And physically and emotionally, I couldn't deal really with that idea.
Yeah.
So for me, research was the better way of doing it because you can be more careful. You can determine what kind of level of risk you can get into. And certainly that has changed because at the beginning, I did things with plants so if you think about ethical involvement there, right? Most people say, well, you're hurting a plant. That's okay, right? Yeah. You mow the lawn, right? So you don't ask yourself, is it hurting? But it is. And any living being will have some of that pain or how it signals. But it's different, right? We're not talking about acute pain. We're not talking about a human being being in pain and things like that. So, but in research presented the opportunity to do that. But I think deeper than that for me was just this need of being able to ask questions. And that's really how far I wanted to go. Yeah.
So being able to ask the important questions and solve these problems that down the line will affect hopefully patient care. And
correct. Yeah. So after doing that experience, that triggered me to want to do a PhD in neuroscience. And so I did do a PhD studying Alzheimer's disease. And at the time, it was the emergence of the idea that you could study all these genes that are associated with Alzheimer's. And it was the research I wanted to do was basically get this new possibility to study all these genetic changes. All these new tools were exploding. Now we're talking about new generation sequencing, right? You can get your whole genome sequenced.
Yeah.
Back then, just having 300 genes done was amazing, right? So you've seen in 15 years how that explosion has happened. Now it's nothing. You can pay $1,000. You get several genomes, no problems. But back then it was like a couple of hundred genes. But the idea, what was fascinating was this idea that we have a blueprint that we get from our parents, and that somehow that blueprint reacts with the environment and instructs our brain of how to work. And the mystery was, how does that interaction change in health and disease? And where does it go wrong? And I was fascinated by the idea that something was changing, that the blueprint didn't fit the environment anymore, right? So it's like, it works fine, except when you're in stress. It works fine, except if there are these other things that don't go well, like you've been taking too much salt or you've been eating too much sugar, and then the blueprint doesn't work. And why doesn't it work for this person, but it works for this person? So these questions were all starting to boil during my PhD, and that genetic interaction with the environment, which is old. It's old as Darwin. Darwin is the one who started with that. The idea that the genes were somehow related to how our diversity happened. That's where it started. But it is now one of the tools we can do. I mean, right? I heard this morning on NPR that they've been doing genetic, there's the first FDA approved genetic therapy for Duchenne muscular atrophy, right? Okay. So that's kind of like a ALS, and that has been treated with genetic gene therapy. It works. That means that we're at the stage where we can correct the genes. That's the blueprint.
Was that the one where they went in I guess they were infants and they changed
Right.
I heard about that as well.
Right. So, but some of them are not working now. So they've got liver problems.
So fix one thing,
Right. So now we're getting into that balance of how and what and ultimately, I guess that from my PhD, I was very frustrated during my PhD, which is half of what a PhD does. It confronts you with the reality of how much work you need to do to answer even the simplest question. It's huge. And I got the little bit of a tidbit of something that, I understood a little bit more about Alzheimer's, but it showed me that what we're doing in research sometimes when you're studying disorders or neurodegeneration or dementia or things like that, we're studying the disease. But sometimes that doesn't tell you more about how it starts. Because to understand how the disease starts, you have to go from a healthy cell to a non-healthy part. And that crossover to go from one to the other you have to be able to define how that healthy is. So how do you define going from healthy to unhealthy?
You have to define the good before you can see the bad.
And so that idea formed in my mind during my PhD that I was done looking at the unhealthy. I had an idea of that. What I wanted to do now is to look at the healthy.
Like see what works.
Right.
So that we can understand if it doesn't work, that's part of the issue.
And then try to push the healthy or use tools in science that you can do. For instance, this is what I can do in an animal model where ethically I'm completely fine with it, using a C. elegans worm, which is a transparent earthworm that nobody cares about because it's super small and most people think it's a parasite. Does it have pain? Yes, it has pain receptors. Does it have a conscious? Probably not. It doesn't have any of the things that are related to higher consciousness or pain things. And so you can add, and it lives very quickly. And there's immense genetic tools. And the reason for the tools is that what I want to do now is I want to go from a healthy neuronal network that works for this animal. It can find food, it can live its life. And I want to know, when does that change from healthy to unhealthy? And I can do that in different ways. You can feed it poisons, sure. But you can do it genetically, right? So you can go and and one of the amazing revolutions of this last 20 years is that you can go and look at, on the internet or on medical parts, you have databases, like for instance, FARI is an autism-based database, and you have the genetics. You have all the patients with the genes that were modified. And the amazing part about evolution is that you can go look at that gene in humans, put that with all the other genes of all the model organism, align them, and you can find the exact same spot.
Like in the model organism?
In the model organism. So in that gene that is concerned from C. elegans to Drosophila to humans to cows to, when you mention it, there are parts of proteins that are exactly the same and have not changed since billions of years.
So much conservation.
That thing works.
Yeah.
So nature is really good at using modules inside things that work and keeping them. And it changes a little bit around and puts them in a different context, and it does a little bit different. And you will see this again and again and again. But that little core thing, if you take that and you change that and you put that mutation in C. elegans, it does the same problem as in humans.
Wow. So that's super interesting to me, because I've always wondered where the link between model organisms and humans were, because we're studying many people won't be familiar with model organisms, and we're studying a tiny 1 centimeter long translucent earthworm. How the hell does that apply to humans?
Correct.
But it's through these genes that are conserved.
The DNA that you have in your cells that we all have, that's the link of all life. And that life, that code, it's not all of it, because as I said, that code has to interact with the environment. And that's that interplay that becomes what we know as life and individuals. Everyone is unique. But the DNA is what we can change, and we have a way to do that now, and to be able to study that, and that's the part that's also conserved. So I'll tell you an example of a friend of mine when I was doing my postdoctoral work in Utah and Salt Lake City. He had finished his PhD, but at the end of it, he started being interested in being a genetic counselor. And so he worked to do that. You have to pass board exams because it's an official sort of federal position. And he was working with C. elegans in a neighboring lab. And he worked with a family that had a girl that had been diagnosed with very severe mental retardation. And she really had really big problems. She can see well, she can talk at all, really bad things. And they identified, while he was there with those parents, they used a new generation sequencing. They find the gene. And it turns out that one of those genes was an acetylcholine release, which is one of the neurotransmitters that we use in the brain to activate things. The well-known one is for neuromuscular junction, so you use it to activate muscles. But the other way of thinking about that is the vagus nerve. This is how you regulate your heart rate.
Yeah.
But it also works as being a tension in our brain. And it turns out that what he found, the gene that was related to that, was actually a gene that he had studied in his PhD.
Wow.
Turns out it was something that had to do with how the vesicle binds to the membrane to release the neurotransmitter. And he took that mutation from that little girl, he made the mutation in C. elegans, and then he recorded, because he was an electrophysiologist, he recorded how that worked. He looked at the behavior of the worm, and he did that 3 months. 3 months later, he came back and told the parents, this is what's happening with your little girl, with the neurons. They can't do this and they can't do that.
So was it like an issue with the vesicles docking and then not releasing in effect?
So she had a massive decrease in the number of neurotransmitter release that she could do.
And would there be a treatment for something like that?
You could try. So the idea now that you identified the site is perhaps to try so one of the possibilities was to try to use some of these toxins. One of them is botulinum toxin or things like that to try to modify that release.
Okay.
It's very tricky, especially in the brain.
Yeah, because there's so many unique receptors and neurons.
But what he shared, what was the amazing part is that now you can do that with this model organism. He could give these parents an answer within 3 months.
Yeah.
And they were not scientists, but they now understood in a physical way what was going on. And that was priceless.
Yeah, I mean, that's super powerful, I think. If you have a medical condition and you don't really know what's going on. A lot of them are super clear. You break your ankle, you know what that means, roughly. But
the brain is mysterious, right?
It is.
You don't see where the break is. You only realize it in the behavior. And there are some super powerful programs at NIH that did that. So, for instance, there's an initiative on rare genetic diseases, and all those mutations are being tested in C. elegans in Oregon. There's WashU's doing a program, and it's a multi-center program where you can test all these things, and it will have amazing impacts on rare diseases, which big pharma can't do, because if you only have a couple of 100 individuals in the whole world that have this,
it's not lucrative.
How are you going to make money out of it?
Yeah.
How are you going to make a medication for it? But if you can find out a little bit more about it, maybe you can use things that have already been done.
Yeah.
Right. You reuse medication. That's the power of the genetics and the models.
Interesting.
Right. And the other power is that you did it in a model. You didn't have to do it in a mouse. You didn't have to do it you didn't have to do it in another child. Right. You didn't have to go in and put someone at risk.
Yeah.
And that's super
That's
powerful.
the power of models, really.
Right.
It gives you the ability to learn about all of these things and then translate them over. So I guess in terms of these genetic conditions, we see, for instance, the acetylcholine vesicles not docking well and not releasing. What do you see the future of treating that? Like, are there avenues, you think, for these rare genetic conditions that we can reverse them?
I think what is being done is the right way forward. I don't think that I have anything that's completely original about this. But I think that what people have been doing is sort of trying to categorize them functionally. So what do I mean by that? So as you said, in this case, this mutation affected the release. But you could have mutations that affect the vesicle forming at all. Or are there mutations that affect how does that neurotransmitter get from where it's made, usually in the cell body, all the way to the synapse? And by categorizing and looking at more and more of these things, we can put them in boxes. And I think that the way people have started to move forward is to say, if we now have medication, drugs that have been researched that affect globally one function versus another, you can reuse them for other things. So that's one way we can do that. And I would put that under one of the super powerful things that should happen now is that we can finally, with the 23andMe and gene sequencing, if we do enough of us and we keep that data, carefully hidden but available for medical aid, if you as an EMT come in and there is someone who's suffering from something and you pull up something on the internet and you have the entire genetics, more important than the genetics, that has been translated to this person has a mutation in here. Do not take this anesthetic. It will kill them. Same thing for cancer drug. Same thing for mental health. Do not take this SSRI. This person has a mutation in this particular process that if you take that thing that you supposedly will help depression, but for that patient, it will make it worse.
I see.
So that power of the genetics with that is the future of how we can do some. The next part will be the gene therapy. That is the next part. And the gene therapy will be in 2 levels. One of them we've already seen with COVID.
With the mRNA?
Correct.
Okay.
The mRNA vaccine technology is what we've been looking for before we can now go into, and this is the very tricky part, right, to say, do we really want to rewrite the human genetic code, right? That is ethically a huge question.
Yeah.
But if you can, instead of rewriting the DNA, affect the mRNA, then you're in the clear.
Mm-hmm.
And the reason for that, so for people who don't understand this, that DNA is translated into RNA, and RNA is the one that gets into a protein. If you can affect that little level of mRNA to protein without affecting the blueprint, you can modify how the cell responds. And if there's a defect of not enough copies, you can prevent that mRNA. Let's say instead of 10 copies, you have 1. But let's say there are drugs out there that prevent that 1 copy from being degraded.
Yeah.
Trick done. You can make certain copies stay longer.
I see. Yeah.
And then you can change that. There are many, many examples.
I could almost see too, you have an mRNA therapy where it goes in and provides an mRNA for, like the vesicle example, back to that because I think it's a good one.
Missing gene,
and the mRNA is introduced, and now it's able to produce whatever the vesicle was missing to dock one of the SNAP proteins or whatever. And now it's there, and now it's able to work, and it's just seemingly solved.
And one of the amazing thing of what COVID did is that it has given scientists in all processes of when we develop a new cure, things sometimes are slow, and they have to be slow to be secure. Sometimes that security is overrated. It slows down things more than what a patient or a family would want for their loved ones, right? That's because they're the only ones waiting for it. But now with a pandemic, everyone wanted that answer. And that's where the calculation of risk and reward comes into play, right? Almost everything we do kind of go into that. And now if we look at where that is right now, like they've reversed that. They say, okay, the vaccine was done during COVID. We did all these things. And naturally what should have happened is that we had to go back and now say, are these vaccine or these things that we've been doing safe for how many people? And what kind of percentage are we okay with that have a problem? And what are we not okay with? And so that decision has to be made at the society, at a political, at all kinds of levels. But for the scientists, it has to be as secure as possible. Right? We don't want anything bad to happen.
Yeah, you want the highest.
But the problem is if you don't test things, you never know. So that conundrum comes into play many, many, many times.
Yeah.
But for the mRNA, one of the things they found with COVID was that what would be your prediction of how long one of those mRNA would last?
I mean, 6 months maybe?
Right. So at the cellular level, an mRNA usually lasts like a couple of hours.
Oh, yeah. No, that's,
right?
That's right. Yeah.
So, but the effect was 6 months.
Yeah.
So that's the part that we're getting into. It's really interesting now, right? So how does that work?
Yeah.
And how
do we know
use it?
how that works? Because you introduce the mRNA and it's there and it builds the spike proteins.
Well, it depends on what you do, right? So in this case with a vaccine, it built something that then the immune system got a print on, and the immune system has a memory. That memory lasts. Yeah. But it depends what you're looking at, right? Then then the devil's in the detail, like almost everything.
Because if the immune system if it's not an immunological sort of thing, and it's just meant to replace one of the missing genes, it doesn't seem like it would last quite as long.
Correct. Yeah. But we don't actually. A lot of those questions are unknown. Yeah. Because one of the things that life does, and the brain particularly does very, very well, is that it's all a balance. And some of the things that we think are basically, we look at it as like, well, there's not enough of this. But you're looking at a brain of a person that has dealt with that problem for already 20 years. So the brain has already made changes to try to compensate for that, right? I was talking about redundancies within redundancies within redundancies.
Yeah.
And that's the tricky part of the brain, is that if you look at what's going on now, you might not be looking at what is the problem. You might be looking at what has compensated to try to make the problem less. So a lot of the things that happen when people have started treating neurodegeneration is that they've made it worse, because what they were looking at was actually trying to help.
It was almost like a survivorship bias where you put, if I'm not sure if you're familiar where the survivorship bias comes from, but in World War II, the airplanes that came back from battle, they would look at and say, okay, they got hit in these places, so that's where we need to put the armor. And it didn't help. They needed to put the armor in the places that weren't hit, because those were the places that the planes that crashed and didn't come back, got hit.
Correct.
So it almost sounds like something like that. You're treating
That's a great example.
But in reality, you're unaware of the actual issue that is being covered up by the compensation.
And the more complex the thing you're looking at, the more the more intricate that becomes.
Yeah, and the brain is like the most complex thing. So
and that brings me to more of what my research is about. So I've talked a little bit about C. elegans. C. elegans has only 300 neurons. It's a hermaphrodite animal. What I mean by that, it's both male and female, right? And so it can self-propagate itself, which makes it really interesting genetically because it makes clones. So from a genetic point of view means that if you have one worm and you change it, then you propagate that change and you can keep it. And the other thing about it is that in 3 days, it goes from an egg to an adult. So it's really fast. And then for me, at least the first time I heard about it, I thought, oh, wow, this is really cool. In 5 days, it has neurodegeneration. So Alzheimer's that takes 50 to 60 years in a worm takes 5 days.
Wow.
Right. So
That's
you don't
so
have
useful.
Right. And for
Saves
drug and money and everything, it's so interesting. Of course, you can ask yourself what happens in 5 days is not the same as what happens in 50 years.
Yeah.
It's true. It's not the same. But some of the principles can be done.
Yeah.
So my research right now is getting really interested in, it started off with just how does memory work? And I was just interested in this one thing that has to do with glutamate receptors, because that is an ion channel that's absolutely required in all vertebrates for neuronal function. So cognition, memory, things that we know about. And all I was asking was, how does that receptor that's made in the cell body go all the way to this other part of the cell and neurons? And neurons really long, with long branches and all kinds of compartments. How does it get there? And that seemed fairly simple, somewhat. But now I'm getting interested in another part, which is I'm interested in the mitochondria in neurons. And I'm getting interested in that because of things of society. Our nutrition is showing more and more that some of the things in our environment are affecting our mental health. And the mitochondria is the great integrator of energy. It's what delivers energy to any cell, but it becomes more acute in the brain. And I think the best example we have of that is that although you can stop sugar, you can basically stop eating actually for 3-4 months or more. Eventually you will die if you really, really push it. And as long as you have liquids, you can burn a lot of fat. And so that compensates. But if you take the example of energy like oxygen, your brain can only go for 5 minutes without oxygen. For most people. Some people can go longer, but usually 5 minutes and you have a problem, right? Your brain shuts down completely.
You need that oxygen for energy.
Where does that come from? And so I'm going back to the original, my grandfather was not a person, then became a person with one chemical. And now I'm interested in how does the food or the energy control of mitochondria, how does that determine who we are? Because we now know more and more how mitochondria control the synapse, which is the contact between neurons that really determines memory. And I'm getting really interested in a lot of how that energy gets regulated, because there's so many things that we thought were neuronal, problems like epilepsy, like Parkinson, that actually seem to be coming from potentially nutrition, microbiome, and things like that.
Okay.
Or at least being modulated. So there's the genetic part. We found the genes. That's correct. There are genetic mutations and genes that strongly affect that. But the cases where you have more subtle epilepsy or more subtle, longer-term Parkinson, how long does it take to get there? That's where vulnerability comes into play. So certain neurons in our brains are more vulnerable to stress conditions, and dopamine neurons are one of them. And there are other neurons like that. They're vulnerable to different conditions of the brain, and that can be either overexcitation or too much sugar or things like that. And I'm really interested in this idea that mitochondria could regulate this. And it's related to this one concept that is also triggered by the AI revolution and the data sets. This seems like jumping topics, but if you ask yourself, how much energy does a data center require? How much energy does a computer require to have memory, RAM memory, or data storage? And then you ask yourself your question, how much energy do we need to have memory? And what do we mean by that? Do we need energy all the time? Does it mean that if you fast, you lose your memories? No, you don't.
Yeah.
but can they come back?
Mhm.
Or in the case of my grandfather, he gave a chemical back and then everything came back. How does that work? Where is that control? So mitochondria could be a way to understand more about how the energy gets put in.
Okay.
And some of it certainly is in development, right? So as we grow as kids and teenagers and adults, those mitochondria change and go to places and get anchored. And then the next level is when we get older, those mitochondria work less and less efficiently.
Okay. And that's where the issues start to come in?
That's where things can be becoming interesting.
I see. So the mitochondria are necessary to maintain memory just for the energy. Your computer.
Well, I'm asking that question.
You're asking that question.
Right. I'm asking. So you know how I told you I want to know how to go from healthy to unhealthy?
Mm-hmm.
So I want to know, when does the mitochondria, if you push, either you deprive it of food or you decrease the energy, when does it start affecting the neuron and what happens? And which neuron is different? So is there a difference in this type of neuron or that type of neuron? And is it because the mitochondria is now past a point? So there are these molecules that people call reactive oxygen species, raw. Mm-hmm. So we eat blueberries, right? We eat all these things. Those antioxidants, or red wine is one thing that people have talked. That's where those things get put together. So I take those society questions and I ask myself, how does that boil down to a scientific question? And the mitochondria for me is fascinating. It's doing all these things.
Yeah.
So that's what my research is about now, is trying to figure out how these mitochondria impact memory and how do they go from a situation where they're doing fine to a situation where they're not doing fine.
Okay.
And can we reproduce this, and can we figure out this for vertebrates? Is there things that we can figure out from our environment or our food or even sleep?
Yeah, I mean, it makes sense. Energy is such a fascinating concept because it really drives like everything. All of our infrastructure, computers, servers. Like you were saying, it's all in energy. It all hinges around energy, and humans are no different. So it would make sense that even neurological conditions would also hinge around energy. And down the line, when you see neurological decline, that the energy producers, the mitochondria, if those start to have dysfunctions, that might start to affect memory and other neurological aspects.
It's a key lynch point, right? Because if you look at what's going on with a lot of what the society is interested right now, a lot of us are interested in food supplements or what kind of healthy foods or things, and all those questions are important for sure. But there are two components of it. One is they're just energy. And if you have enough energy, given that, why would you see a decline at all?
Mm-hmm.
Well, the question might not be, is it the energy, but is it what comes with the energy source?
The other stuff.
There are toxins, microplastics. What are things that, together with your genes, right? The two are related. So that's the tricky part, is that for one person, it would be this. For another person, it would be something different.
Well, it's the constant balance between nature and nurture. You have the genetic code that you're built on, and that determines a lot. 50% of personality even is determined by your genetic code. But you have that other half of environment, and that also dramatically affects you have the foundation, but what you build on that foundation is going to be determined by non-genetic factors a lot of the time. Yeah, and that's what makes all the research interesting, right? So how are we going to be able to change that with behavior things? There's so many really interesting things coming up, right? There's the genetics that we can now do with the mRNA vaccines. There's the fact that we have access to the codes that, we could now inform, medical doctors, EMTs, everybody's like, if somebody had secure access, because it has to be careful, right? That's going to be a big issue. That's the major problem, is that it has to be secure, and that, that people can then say, before I even do anything, I don't want to, you can destroy a lot by doing the wrong decision, which in the medical field is quite, obvious, but that's, if we know from the genetic code, if it helps us to get there, then it's a huge help. Yeah. And then you'll be able, not only will you be able to use the genetic code to tell you what not to do, but it'll also tell you what to do. We'll probably get more narrowed in, like specialized medications. And, this medication might only work for 30% of people, but you pull up their genetic code and see if they're one of the people that it's going to work for. Right. And the more we can also, I think there's also a lot of interest here
with the genetic code to kind of do, if there's ways to do experiments where you can repurpose medication. Some of the coolest stories that come up in the last 20 years are things where, let me see, one of the drugs that a friend of mine was actively looking at that was basically for erectile dysfunction, was the blue pill called?
Viagra.
Yeah. And so they looked at that, and then they noticed that, they used that for that, but then they noticed that that substance actually had a huge effect on headaches.
Interesting.
And so they looked, and it makes sense. It's a vasoactive dilation product.
Yeah.
But it doesn't only work in one part of your body. So when you take medication, right, you put it in the blood flow, it goes everywhere.
Yeah.
Right? And everybody's just like, okay, well, you take ibuprofen, it works here. Well, that's what you see, but it's working everywhere.
Yeah.
And that's the part that's interesting, is that if you could target it or change that slightly, which is what a lot of pharmaceutical companies do, is now, can you take that same molecule, change it a little bit, and now it will act in your lungs. So for your lungs to breathe and open up really well, you have to have vasodilation.
Yeah.
And that's one of the things they saw. They saw that it had an effect on headaches. They said, that sounds like something that's working in the blood vessels, which makes sense. They even went looking at where else it was acting. And they had a potential second patent right there just to help people with cystic fibrosis.
Jeez, that's really cool.
Right?
Yeah. I mean, it seems like a much more economical way as well. Like, you already have this drug that it's a vasodilator. So let's see if we can tweak it and have it vasodilate in other areas of the body. Or the other one is Cialis, another ED drug. I think I've heard that one they're finding that it's good just for guys to take when they get older, to have like a low dose Cialis to help prostate health, just again to increase the blood flow.
The blood flow for different parts of your body and things like that. Yeah.
We're
going to find, I think there's a lot of these things are incredibly interesting. And I think the combination of our ability to now use the genetic information together with this, and potentially, I think if you put that in the mix, the AI mix, particularly if you use the AI the way I think it should be used, which is it's incredibly powerful to sift through billions of data points with a very, very detailed way of looking at patterns that our brains are not good at. And it does it very quickly and efficiently. And if you use that to look at, for instance, genetic code with medication, you could find amazing things. Really. The potential to combine things with this massive amount of data to kind of try to recombine things or use that is amazing.
Yeah.
And but the fact is that we need to get into how can we do that, right? How can we chase that? So the themes that are really interesting here is, nutrition is really important for your brain, and what does it do? How does it do it? Is most of what we're talking about for neurodegeneration. So a really cool question in neurodegeneration are things like, are we active too long? Is it the sleep? Or is it a combination with our food? Or is the energy with the mitochondria? How does that work? And if I could test that in the worm, right? If I could deprive them from sleep, change their food source, change that, show that the mitochondria changes, and then most important of all, I then put back one piece at a time, reconstructing the toy that I broke, trying to make it figure out how it works. If the molecules, and the molecules are the same in vertebrates, then I can go and say, okay, you guys, this part of the protein works in the brain in humans or in vertebrates, mammals, cats, dogs. Why don't we try it?
Yeah. Is that what you're doing in the lab, is like, depriving them of X, Y, and Z and seeing okay. Very interesting.
Right. Yeah.
So
I'm also modifying the mitochondria itself directly. I'm playing with something that has to do with, so when neurons get active, there's lots of calcium that comes in. It's known that calcium can be toxic, and that's also something that, so for instance, Alzheimer's, there's this phase that goes silent a little bit, but is now coming up to the forefront, where it seems that there's certain circuits that are hyperactive, and then they go silent. And so it's thought that this first phase of hyperexcitability is where toxicity starts. And I'm really interested in that idea because I think there's a lot of things in our society that show that, we don't get as much rest. We have screens. We have a lot of things that where basically our brain is active a lot. And people have seen already that restful things, like we're talking about nature bathing, we're talking about, but essentially it's all things that have to do with how do we calm things down?
Yeah, that makes a lot of sense, actually. I haven't really thought of it that way. But even if you do get sufficient sleep, if you're staring at your phone 8 hours a day, getting like constant stimulation, that's probably more stimulation than we were built for. So
right, and we're attracted to it, so it's really difficult to do. And I think that the changing. So the other part of really interesting, and it's always harder, is to think you can change like the hardware, but you can also change the software if that's the behavior.
Yeah.
Excuse me. And so I think that we all know that we could do it, and how much can we do? So the best way is if we can do both.
Like change the, our behaviors
Behavior and
yeah.
And I would say that probably easier in some ways, that's where science and policy and other things come together, is if we can change the behavior that lead to a healthier choice by default.
Yeah.
Right? You don't have a choice. You only have healthy things.
Yeah, no,
So
that would be good. Less like it does seem a little ridiculous, the amount of unhealthy options that are just right there available to us. But it is a tricky kind of thing to navigate.
And it's all trial and error, right? I mean, we live short lives compared to the length of the globe and things like that. But I think that if we can allow this to happen, and we have to give it a chance. And so C. elegans is another example of how you can do it, right? So one of the things that I find interesting is that thing lives the life cycle is 3 days. So you can go through 100 generations in 1 year, right? So that tells you how quickly I can try to change things. And I like that idea that, and some of the things that I think are the coolest is, one example in my lab is that after years of using what we would call a control strain or wild type strain, suddenly stopped being what we would call wild type. And so that's that part where I told you, right, there's the healthy and the unhealthy. Where when you do experiments with genetics, you use what you call a wild type, which is supposed to be the pure genetic animal from when you took it from the wild, to something that you've genetically altered. And you compare them. And you analyze the result by saying, this one's different from that one. And that's how you say there's something that you can then follow. Now, what if your wild type or your healthy is now longer healthy?
Yeah. Well, I mean, I could almost imagine in those 100 generations, you could start to see genetic mutations and changes like to adapt to the environment. And I could also see how if you introduced like certain negative environmental stimuli, like, poor nutrition, and seeing how those change over time, you could almost extrapolate that out to humans and say, well, if we continue with this sort of negative diet and environmental toxins, what's that going to look like 100 generations from now?
Correct.
We can kind of postulate that with what you're doing.
Yeah, you can see that happening in the long term. And then there's another thing that I find fascinating is that, so I told you wild type. What I didn't tell you is which wild type. So there's one from England and there's one from Hawaii. And the one from Hawaii is not the same as the one from England.
No.
Right? And so now, which makes it even more complicated, if you have people coming, let's say, from Hawaii to America, or the U.S., or from China to America, or from Switzerland to China, do I compare myself, if I go to a medical doctor, to people from China? Is that the reference?
Yeah, like how much
would what would they pull on to try to figure out that I'm healthy versus they're healthy?
Yeah, like how much of our just environment that we live in determines what's healthy and what's not.
Right, and our backgrounds of where we come from. So, we tend to think that, yes, we're all equal. We are, mostly 99.9999%. But then that little bit of a difference makes, if you were in Hawaii or in Pacific Islander, where your genotype has been accustomed to every now and again, and this is kind of what they're thinking about diabetes too in those populations, is that they're prone to that because their metabolism was incredibly good at storing fat, right?
Yeah.
They could go on for a long time without food because they would island hop, or at least that's the idea, right? They don't have the same issues we do with diabetes and fat and food. Not at all.
Okay.
They could eat fat and not have a problem with it, or we could eat sugar, but they can't eat sugar. So now the environment, this is coming back to the question is, what is the healthy? What is that?
It does depend on the person, it sounds like.
The context, the places, and all these things. It's fascinating questions of how, now we're going to have to put like different questions. So Yeah, I love all that thing. I think it brings back so many questions of how the organism works, how the biology works, and can get boiled down to basic medical questions, right?
Mm-hmm.
Do you need to give, let's say you have somebody comes and there's, somebody's been shot and his Asian Pacific or more like European, Caucasian or Mexican or Indian, Native American Indian. Let's say the genetics would be different and that the dosage you would give from the anesthetic to this person would be completely different from that person.
Yeah, I haven't even considered that level of like, down to the region you're from, like the microgenetic differences.
We don't know.
Yeah.
I mean, I don't want to categorize those, but I think that what is fascinating is we're seeing a mix of things, and now it's going to be really down to, I don't think we can categorize that anymore because the mix is intense.
Yeah.
So now we can, but now it's no longer good to do that. Now I think that unless you have the genetic code, there are things that you can't tell.
Yeah.
Absolutely.
Oh, yeah.
Right.
But I mean, the genetic code just makes it more individual and personal
Right.
when you boil it down.
And I think that the right word here is personalized medicine, which is what the concept is absolutely correct. And
Yeah.
I think that I have a lot of hope for how that's really going to work out. I just have concerns about how good we are at keeping that information where it should be.
And just the ethics of it, and
Right.
What do we use it for,
Right.
especially with the genetic side of things.
I think it would be really cool if we could like create like banks, like DNA, right? If you can put your money in a place that's supposed to be secure, could you put your genetic information in a place that is all secure? That is yours, like this is what you have, and then on necessity or in places you can share it.
Yeah.
But only in those times.
Yeah. It's going to be tough though, right? Because with the, increasing level of complexity with AI and coding and eventually quantum computing down the line. I mean, all of this stuff is going to be stored digitally, I presume. And anything stored in a digital way is going to be prone to
you can access
malicious, like hackers and so on. And
at some point,
and it's tough, but we're going to have to figure it out because it's probably inevitable.
I think there's good ways to think about that. I think we're on the brink of thinking about that in terms of where we're going. But so I mean, the C. elegans is what is cool with that particular model is that now I'm, to give an example of how computation and AI can get into it, I'm working with someone from San Diego that we're now doing so C. elegans is fast, right? And you can do the experiments, but one of my hobbies is science fiction. The reason why I like science fiction is because I can imagine situations so much faster than I actually do the experiment.
Yeah.
And now I've been starting to work with somebody who does computational modeling. So using computer equations, mathematical models of signals to go to that next step. Can I go faster?
Yeah.
Or can I do more experiments to make sure that the experiment that I will do physically is the right one?
Yeah.
So that I don't lose time or waste resources.
Yeah. Giving you like a pathway, a theoretical pathway.
So you can test things virtually and then get the best educated guess and then try those out.
Yeah. What's your favorite science fiction series?
So right now I really like a series called David Brin, the Uplift series.
I haven't heard of that one.
And there's another one that I love is from Colorado, Dan Simmons. He wrote Hyperion and Endymion.
Okay.
I think there's something being made with Hyperion. And I, as a younger student, I loved Asimov.
Yeah.
All the AIs, all the iRobot and the laws of robotics, and those were foundational for some of the things.
Foundational, yeah. Yeah, no, I'm just getting into science fiction. I'm making my way through Dune right now. And
You just mentioned my other biggest favorite. Dune is a little bit of a different one because it's science fiction, I think. For me, it's a political science fiction.
Absolutely, yeah. You read it, and I mean, there's sci-fi stuff. There's space travel. There's like, cloning and whatever else, like long lifespan. But it is more like a political, almost like a piece of literature masquerading as science fiction. But the sci-fi aspects of it are really, really interesting too.
Oh, they're cool.
Because I think they're like they were very forward thinking, like the Butlerian Jihad, the war against the machines. It's like, that's pretty good for the 50s, and the genetic stuff they do in the books is
they're really good questions and concepts that we should be toying with. And there's all that. And one of the things I always wondered, and one of the things that maybe you will see, at least for me, so Asimov is the last utopian science fiction.
All the other
Almost
ones are
all the other
dystopian.
one are dystopian.
Yeah, that's so interesting. I haven't read Asimov, but he's like next on the chopping block for me.
Right.
I didn't know that, but
At least for me, that's
yeah.
what I mean, I'm no expert. So
Yeah,
so
but no,
don't
I mean,
quote me on
I've read
it.
other sci-fi, and yeah, none of them were particularly like, yeah, we're gonna usher in the utopia. I think everyone is like, okay, we need to be really careful, and I think fiction is probably the best way to explore that because you can't test it in a lab, and it's human ethics, human morality that you're talking about.
I think, but so I want to circle back to that. I think that some of the things that we have right now, and I guess one of the things that's driven me to come to America as opposed to be living in Switzerland was the fact that I had the impression that lifestyle in Switzerland and level of living are really high, so people have a lot of things. But I felt like in science, that was a negative because a lot of my colleagues or benchmates spent a lot of time worrying about things that could go wrong. And a lot of worry was losing what you have. And in science, if you don't take any risks, you don't ask any good questions. So a lot of the questions became incremental. They're not really worthwhile.
Yeah.
So, and I think that's another thing about what we need to be careful about right now is we should not be afraid of asking bigger questions. And it takes commitment and this sort of brave sort of new frontier idea. And also I think that I'm a parent now and I have to tell my children about what's going on in the world and things that are, good or bad. And I realize how much of an influence that is, of how much by the lens of what I'm looking at, do they react in a way that becomes more negative or positive? And that's, first of all, it's very, very scary.
Yeah.
And second of all, it also kind of it shows us that if we now looked at what we have, so let's take all the points that we took together, like personalized medicine, mRNA vaccine, mental health, all these things, is there a lot of reason for hope? There's a huge reason for hope. There's lots of really good things here. Are there lots of reason for concern? Lots of reason for concern too, right? But I think one of the things we have had a tendency to do is kind of like overburden the parts that are dangerous. And we're warning, and this is bad, and that is bad. But to be honest, we need to change. We need to have, for those of us who think that climate change is real and how that's really working, we need to have a positive attitude to we can change this. And we can. We can be brave. We can move the things and the needles. And the science has a method that goes through specific things, that tests, that gives you answers that step by step can go forward. But there's also two-speed science. There's the one that never takes any risks, and there's the one that does. And I think for me, at least in my head, COVID is a huge example of what that was. Somebody took a risk on the COVID vaccine, and that was huge. The number of people we
Such
saved
a big difference
with that was enormous. That shows you that you can't always be careful on every possible thing. You have to have some big ideas.
No, I totally agree. I think it's so easy to get lost in the negativity of everything. And I think damn smartphones don't help with that. We have a negativity bias and our attention gets locked onto negative things. So it seems to inflate that sense of negativity. But I think there is a lot to be hopeful about. I think there's a lot that we can and are doing to focus on these big issues like climate change or looking at, the pandemic. Like there was a lot to not be hopeful about with the pandemic. But I think when the chips fell, especially with the vaccine, like we got that done super quick and it saved so many lives. Like there's a lot that we can do if we just have the bravery and hope to actually change
and some
them.
of the things we work on, I mean, it's always been this dichotomy in science, right? I mean, is nuclear technology amazing? It is amazing. Is it dangerous? Oh my gosh, right? And for some of the things we're doing with genetics, same thing. Is it dangerous? Yes, it is very dangerous. Is it very promising? It's amazing. We could do amazing things with these things, right? Like with great power comes great responsibility. That's the same idea, is that if we do this well and with the aid, I mean, this is one thing I say that AI and some of the databases are amazing. This accruement of knowledge allows now to intertwine things to find solution at a level that is unimaginable compared to 100 years ago. But it does also mean that we have a choice where to apply it and what to apply it to.
Yeah, and that seems to be the biggest question
Right.
is with AI in particular, I think,
Yeah.
is we have this superpower now, and what we use it with or what we use it for is really going to make all the difference, because it could easily be used for malintent, and it'd be really good at that because it's very powerful, and it could also be used for amazingly good things like
we
genetics,
can make so many
breakthroughs.
discoveries. I mean, the Nobel Prize, the recent one, was done with AI doing for
AlphaFold.
protein, right?
Yep.
AlphaFold. So that alone, the ability to kind of go into what we can change and how that's going to have repercussions for science and health and things is immense.
Yeah.
One of the things I do want to kind of get back into, and one of the things I think is really important in science, though, is although all these new technologies are really important, one of the things that I think is really very important for your generation in class, and one of the things I like to have students, is one of the things we can't skip in terms of the way your mental process learns about life and complexity and experiments and things like that, is the part where we learn critically about what is real and not real, and what works and doesn't work, and what processes can be used. We can't use AI as a crutch.
Mm-hmm.
And I think the phone is another way. Is it a crutch? It's a crutch. Can we still remember things? Yes, we should. Can we remember phone numbers? Can we do things? Yes, we can. Are there some that have forgotten how to do that? Yes, they have.
Yeah.
So we have to be aware that some of these things are going to enable and also disable.
Yeah.
But it's really important to me, for instance, that the graduate student and the students in the classes in my lab are also vetted against time-honored sort of like old school things.
Mm-hmm.
And there are certain things that you cannot replace. In other words, if you use a phone as an adult, you have all your years of having used computers and answers and questions to know how to use it.
Mm-hmm.
And you still know how to write, you know how to calculate by hand, you know all this stuff.
Yeah.
And it informs how you think about the questions you ask on the phone.
Mm.
But if you don't do that, if you've skipped all those steps, how is that going to look?
Yeah, because you could almost imagine, I mean, at the end of the day, these tools, we should ideally be using them for the betterment of humanity, the greater good, or however you want to define it. And it seems hard to imagine if you grow up living in the technological space and it would feed in on itself. The questions you ask, the things you pursue are purely online, and that isn't for the human. It's contained only in the internet, I guess, is what I'm trying to say. And
right, there's a component of reality.
You have to have the human experience, the realities, these things that, might seem a little silly, like memorizing phone numbers or, going out and doing science experiments. As a kid growing up, I would do like silly little stuff with dry ice, stuff like that, to interact with the natural world, to know what to ask and what to use the tools for.
Yeah, there's that. And there's also the time component, because digital age has accelerated everything, and our brain can cope with some amount of how fast it can come with it. But it's very clear for AI, for instance, there's no comparison.
Mm-hmm.
Right? If you use one specific task, a computer will be better than our brain, because our brain is not a computer. Mm-hmm. It does not do completely specialized tasks at a really, well, it does, the ones that are unconscious, not the conscious ones. So vision, work, you can catch a baseball in a split microsecond, things like that. Mm-hmm. It is incredibly good at that, hand-eye coordination. Mm-hmm. We never talk about those as being like our computer brain, but that's the computer brain. Yeah. That's what our brain does.
And it makes sense that those are the things that we are nowhere close to making robotics. Like my example I always use is plumbers. We could start replacing surgeons, doctors, these sort of super high specialized things with AI. I could see it. I don't know if people are going to buy into it very much. I don't know if someone wants a robot doing their surgery, but a plumber, how far off are we from, a machine that can come into your house, open up your toilet, fix it, fumble around with some pipes? So far. But anything online, the super, super specialized stuff, those seem to be more susceptible.
Yeah, so the generalization of our knowledge, and the other part, the part that I'm really interested in too, is we used to say that CPUs had a clock, mm-hmm. right? How fast can it go through information? And quantum computing has exploded that in a completely different way. But here we come back to a component you were saying, like you're using the phone or learning. One of the critical things about our life now is the importance of understanding what we do in the 24 hours and in an hour. If we compare that to digital life speed, it's so different.
Mm-hmm.
It makes it really difficult for students or younger children when they look at how fast things evolve, then have to sit down and then be confronted with how slow time is now.
Yeah.
And that is very hard on trying. The reality will not go faster. So there's that, we're talking about science fiction as a way to accelerate.
Mm-hmm.
So in the ideas world, you can go at light speed.
Yeah.
But you still have to confront it to reality. So one of the examples I give my graduate students is like, why am I not a nuclear physicist or an astrophysicist or even a philosopher, which I really wanted as a kid. I really liked philosophy.
Mm-hmm.
Well, one of the things that attracts me to science, biological science, and it could be any physical science, to be honest, is this confrontation with reality. Is that you can go around in circles forever in the intellectual space.
Oh, yeah.
And never figure out if you're right or wrong, ever.
I love philosophy too. And, there was a period of time where I was like reading the philosophy books, reading the philosophers, and you start to lose touch a little bit because, you can just, this philosopher has these axioms and they work in this system and this one has a different in this system. And like, well, they don't work together. So which one's right? Well, the lower axioms within the lower, what's your metaphysic? And so many things to consider. But yeah, I think it seems like where you're going is pulling out of that and just being present and experiencing the reality.
And reality and science has a huge component of that. And that's the part that I don't want to stay what we would call in silico. So I was talking about modeling. Modeling is good, but only if you confront it to reality.
Yeah.
Only. Because all it takes is one deviation to go a little bit further, and you will realize when you confront it to the reality that it won't work.
Yeah.
And that component of checking yourself, making sure that the model or the ideas are working as you think it does here and everywhere. Let's say you test it here in Colorado, but you have to test it in New York, and you have to test it in Geneva and somewhere. But you have to make sure that it's real. That component was what kept me in the physical sciences, because it felt to me that in nuclear physics or in other realms of like really smart math, like math is even worse in some ways, there is no way to test what it is that you have found. And sometimes centuries go by before even anybody can test.
Or even find a use for it.
Right.
Math is hilarious for me. Mathematicians, the PhDs in math, they're researching different kinds of infinity and how they work together.
Right.
And I'm just like, why? Kind of. I mean, it's new knowledge of where, math is kind of a philosophical system. So they're just starting with first principles and building up and up and up and up. But again, where does that come down to
the reality?
Yeah, because humans don't play with infinity ever.
It's so difficult to tell, I think. For me, that grounding in reality is incredibly important for my mental health.
Me too.
But and I know that there are people who don't need that. But I think that for the physical sciences, something that needs to come back to it. But it's also incredibly difficult to predict certain things that seem completely absurd, but then in reality are not.
Right? Like what?
Well, let's take the example of CRISPR. Right, CRISPR technology. The origin, funnily enough, there are 3 Nobel Prizes on DNA, the technology that allows us now to manipulate DNA. CRISPR is one of the last version of that. The version before that, there's a company called NEB, but it's called New England Biolabs, and it had to do with DNA restriction enzymes. They cut DNA. Allows you to cut at certain spots. We even do criminal cases now using that. So is that, where did that technology come from? From people who were interested in bacteria. Were they interested in bacteria that were dangerous for human health? They were just some kind of bacteria, like some slime that was somewhere, right? That's like, you look at it, you go, yeah, nobody wants to touch that. I'm going to look at that. And they found that the molecules in that thing, so CRISPR comes from the idea that bacteria were making warfare two types of species would exchange these parts where the DNA enzymes, that are the CRISPR enzymes, go in to kill the DNA of the other bacteria. That's what CRISPR was doing. It could recognize sequences in the other bacteria and cut it to pieces. Did people pay to look at bacterial warfare from the NIH? Yes, they did. What did that end up doing? We now have the ability to do gene therapy for ALS.
Yeah.
So it went from, I'm looking at the slime on the piece of rock on the pond, to I can do that. Another example of that, for instance, because we're close to Yellowstone, the Great Prismatic Spring, the one thing that has exploded that allows us to look at genes and everything is called PCR, polymerase chain reaction.
Yep.
That came from looking at something that looks like a geyser. Mm-hmm. And people were looking at it going, why are you interested in that?
Yeah. Well, maybe something's living in there.
Right.
Maybe we can get some out of that.
Well, the question was, look, they're living things that are living at 200 degrees Fahrenheit. How in heaven's name do they do that? Mm-hmm. How do they do that? How did that work? That's where we found so many cool things.
Yeah, I think it is so. I mean, that's another just inspiring thing, I think, for anyone going into science is you might be looking at something that you think might not be significant, but 10 years, 5 years down the road, it might explode into some huge thing.
Yeah, keeping the eyes open is like, you can be incredibly specialized in something. And yes, there are lots of symptoms of ivory tower science, lots. But it's difficult to take him off the map because every now and again, some of those ivory towers have come from the tower to now the entire metropolis, right? It is not just, and how does that happen? Is it happenstance? Is it, somebody who looked and applied? How all that combination works, right? People have to talk to each other. People have to communicate. There's a whole bunch of things that come together, and that's fascinating.
Yeah, and it seems like there's no way to tell. Like you can't say, well, okay, this research is going to be the thing that leads to.
Well, you can tell good from bad research.
That's true.
So I think, can you tell which good research will eventually have a huge impact? That is almost impossible.
Yeah.
But good and bad research are very different.
Yes.
And the component of reality check is one of them, right? Can you reproduce it? Can it be applied? Can it be done?
All the replication crisis and all of the different.
The replication crisis is a good point. Yeah. So the replication crisis is also due to incremental science. And that's a part of trying to find things that are small changes that we're trying to replicate. And that half, I would say, of the replication science, the problems that we're having is the fact that we seem to be running out of really big problems that we can tackle, which I disagree with because I think there's lots of big problems we should tackle. But I think that the small problems, when you have little changes and there's lots of little changes everywhere, well, that's a really hard problem because you can mistake something that looks like a change for something that's actually just chance.
Yeah, like if you're studying a thing and it's so small that the change itself could be a rounding error, then it's going to be pretty hard to replicate that.
And I think there's enough of that we've tried to do that we're not doing correctly. And that may be, for instance, a place where AI could help with details that have enough accumulation to actually find patterns.
Yeah.
Where we're not good at that.
Looking at all the data and seeing where we might be going wrong.
Right. And then coming up with a way to test it. Yeah.
Yeah. So what's the future for you look like? What are you hoping to do with research in the next couple of years? Or just career, anything?
Well, I hope to really, I'm making some really good headway with my students in understanding how we get these crucial pieces, these ion channels, glutamate receptors, to go where they're supposed to go. And understanding what are, the genetics and the environmental conditions that regulate that and change our memories or changing forms of memories. So that's one. The second one is this big thing we talked about, which was the mitochondria. And the question really of how does energy regulation modulate or determine, learning and memory and basically retention of memory? And where does that regulation of energy and memory how does it go from healthy to unhealthy? And that's where my future in research is. Certainly here and wherever I can find funding.
Yeah, it's always a big thing.
And I'll do that with collaboration. And and that's what I want to do. I don't want a big lab. I like the places here. I really like the students at CSU. I like the teaching. Teaching always reminds me, because every now and again there are students like you or others who come back and ask questions that remind me that things that I thought were, a textbook or a sentence that I thought was in stone is not in stone. And that dynamics of being confronted with new questions, with new students, new minds, that's what keeps me, interested, curious, and also active. And I think that's invaluable. That human discussion, which I think is incredibly human, is what brought me to science. I had this, I would call it an epiphany, because I think that everybody's had it at a point. But, every human being is a unique collection of knowledge. And all we have to do is open up and ask, what do you know? What are your questions? And if we exchange things, something amazing happens. And that part, I'm tearing up, but I'm really passionate about that, because I think that we've forgotten how much each of us knows, and we keep them to ourselves, and they form opinions, and we confront opinions. That's not how it works.
We have to confront other people. Not confront, but
we have to
interact.
exchange. And that means opening up. And no matter what. And that in science and in anything else, because of course you can extrapolate, but that part of trying to know from new generations and learning myself, that's the future. That's how we get where we need to go, to learn from each other.
Yeah, the spreading of knowledge. I love it. Dr. Hoerndli, you are amazing. Thank you for coming on the show. I love the philosophy that you have. The work that you're doing, I think, is great. It's going to have great impacts for health and human flourishing in the future.
Thank you.
Yeah, of course.
Thanks for letting me be here and talk about what I love to do.
Absolutely.